Have you ever wondered how complex life really is, even in its simplest forms? It's a question that has intrigued scientists for decades. Let's take a journey into the microscopic world of Mycoplasma pneumoniae, a bacterium that was once thought to be simple. However, when a network of research groups dug deeper, they discovered a reality far more complex than they had anticipated. They examined this bacterium at 3 different levels: its transcriptome, metabolomy, and proteome organization. What they found was a level of complexity that defied their expectations. Similarly, when scientists stripped down the bacterium Mycoplasma mycoides to its bare essentials, they were left with a surprising number of essential genes, many of which had unknown functions. These discoveries challenge our understanding of life's complexity, revealing that even the seemingly simple organisms have layers of complexity that we're just beginning to understand. So it seems that even what we consider simple life is not so simple after all. Let's delve into the intricacies of Mycoplasma pneumoniae, a bacterium once thought to be simple. To unravel the mysteries of this minimal cell, researchers approached it from 3 different angles, each seeking to map a distinct aspect of its inner workings. The first team embarked on a journey to decipher the bacterium's transcriptome, identifying all the RNA molecules produced from its DNA under varying environmental conditions. This is akin to reading the language of the cell, an essential step in understanding any form of life. Simultaneously, another group of scientists were busy defining the metabolic reactions occurring within the bacterium, collectively known as its metabolomy. Like the gears in a clock, these reactions work in harmony to power the cell, allowing it to respond and adapt to its environment. Meanwhile, a third team was identifying every multi-protein complex the bacterium produced, thereby characterizing its proteome organization. Proteins are the cell's workforce, performing a wide array of tasks necessary for the cell's survival. What they found was astounding. At all 3 levels, the bacterium was far more complex than initially thought. Each layer of its biological architecture revealed a depth of complexity that defied its label as a simple organism. Thus, Mycoplasma pneumoniae, despite being a minimal cell, showcases a level of complexity that surpasses our expectations. It's a fascinating testament to the fact that even the seemingly simplest forms of life can be exceedingly complex, intricate, and elaborate, challenging our understanding of what it means to be a living organism. The story doesn't end there. In fact, it gets even more complex. Let's take a journey into the world of Mycoplasma genitalium, one of the smallest free-living organisms on our planet. This microbe, with its mere 525 genes, appears simplistic on the surface. But don't be fooled. A group of bioengineers led by Stanford's Marcus Covert took on the challenge of modeling this bacterium on computers. What seemed like a simple task turned out to be an enormous undertaking. It required a cluster of 128 computers, running for nearly 10 hours, just to generate data on the 25 categories of molecules involved in the cell's life cycle processes. The complexity of this simple microbe was astonishing. It necessitated 28 subsystems to be individually modeled and integrated, and many critics complained that their computer model was only a fraction of what will eventually be required to consider the computer simulation a realistic simulation of life. The depth and breadth of cellular complexity in this tiny organism were almost unbelievable. Indeed, even the simplest microbe in the world is not so simple when we delve into its cellular processes. In 2016, scientists embarked on a journey to understand the minimal requirements for life. They turned their attention to Mycoplasma mycoides, a simple bacterium. Their mission? To strip it down to its most essential genes, and in doing so, uncover the bare necessities for life to exist. This was no easy task. It involved systematically eliminating anything the bacterium could live without, a process akin to microscopic surgery. As they worked, the scientists made a startling discovery. The bacterium was pared down to 473 genes. This was a surprising number, far higher than they had anticipated. It illustrated just how many genes were essential for life to continue. But the surprises didn't stop there. Of these essential genes, 149 had unknown functions. These were genes that science, in all its wisdom and advanced technology, could not yet account for. It was a revelation that left even the most seasoned researchers unsettled. Alistair Elphick, a bioengineer at the University of Edinburgh, described the finding as unsettling. Elphick also stated, "If we're already playing God, we're not doing a particularly good job of it. Simply streamlining what's already in nature doesn't seem very godlike, and if anything, is a very humbling exercise." Craig Venter, who led the study, stated, "We're showing how complex life is even in the simplest of organisms. These findings are very humbling in that regard." Moreover, this 473 genes was not the universal blueprint for life. Had they started with a different bacterium, they would likely have ended up with a different set of essential genes. Life, it appears, is complex to its very core. Even the simplest organisms are more intricate and complex than we ever imagined. It's a humbling reminder of just how much we still have to learn about the world around us. In conclusion, despite an intense 70 years of research since the Miller-Urey experiment in 1952, how life can arise from non-life through naturalistic processes is much more of a mystery today than it was thought to be 70 years ago. As synthetic chemist Dr. James Tour stated in 2019, "Every year we understand more about the complexity of simple life. So we are more befuddled now than we were in 1952.